Okay, next speaker, it's a pleasure to introduce Professor Ullas Kothur Seetharam, who's a professor, who's professor at TIFR Hyderabad and Bombay, as well as the director of CDFT, as well as NIAB in Hyderabad. Ullas is very well known for his work on metabolism and physiology, and at the systemic metabolism level, lots of wonderful work on looking at overall metabolism and regulation responses in terms of feeding and fasting, anticipatory feeding responses, and so on in liver, muscle, and kidney. And also lots of mechanistic studies which are at the cellular level, looking at mechanisms of nutritional responses, mitochondrial energetics, mitochondrial outputs, chromatin regulation through sirtuins, and much more. So very broad, versatile range of studies across scales in metabolism, and so it'll be a pleasure to listen to what Ullas is talking about.
Yeah, thanks Sunil. We didn't make a deal to introduce each other. It so happens. Yeah, I guess I'm listed as one of the organizers, but I'm just a name on the board, and I guess it's been wonderful. And of course, I keep coming to CMC, but I think I should visit CSCR more often from now onwards. Okay, so I'm going to talk to you about something about the importance of metabolic orchestration. And I have multiple loyalties, so just as a disclaimer, my lab largely functions in TIFR Bombay and TIFR Hyderabad. I'm going to introduce a little bit about the Arumda, which we have set up as one of the first research centers to study metabolic disease and aging as a concerted effort. And of course, I am administratively associated with the CDFT and now National Institute of Animal Biotechnology as well. Now, for people who are both basic biologists and clinicians, all of us understand the importance of phenotype, right? So phenotype is of course a culmination of both genotype and environmental inputs. And largely we have thought about why genetics becomes important, but more and more we have come to realize that environmental inputs are equally important in terms of driving both the onset and progression of several diseases. Now, classically, I was trained as a molecular biologist, and we were told to believe that if you understand genes, you could solve diseases. Of course, we have sequenced many genomes, and yet we are far from understanding why disease happens. So the classical paradigms in biology was that if you understand genes, you will be able to address everything, and that metabolism is something that was downstream to molecular functions. But turns out that actually metabolism is the one that dictates pretty much what happens even at the genetic level, and this is something that I'm going to briefly talk to you about as well today. Now, come to think of it, this has huge relevance in everything that we would like to study in terms of stem cells, differentiation, iPSCs, and also have implications for cell and gene therapy. Now, biologists are extremely scared of maths and numbers, and in fact, in PhD interviews, if you ask a student, can we ask a simple math question? They said, sir, but I left math in 10th. So, therefore, biologists are extremely wary of looking at energetics and definitely not at thermodynamics, because you can't ignore thermodynamics. Most of the molecular functions happen at thermodynamic equilibriums, and then it's important to therefore address these things. Now, just because I'm talking in this particular conference, metabolism is extremely relevant no matter which way you think about it. It has a huge impact in how your cell and gene therapy mechanisms can work. In fact, whether it's you're talking about CAR T cells, CAR macrophages, or any other immune therapy based modulations, metabolic features of these cells become extremely relevant in terms of efficacies of interventions. But you also have a huge application of cell and gene therapy for metabolic diseases, including for liver diseases and muscles and several others. So if you want to really think about in a comprehensive manner, while you need genomics and of course CGT, but all of these cannot happen efficiently unless you address metabolic underpinnings. Now, metabolism is not something that is a set of unique pathways that are boringly taught in our undergrad, and definitely I guess is also the case in medical schools that it's one of the most boringly taught subject. It is a set of pathways that actually provide meaning to pretty much everything that happens inside the cells, and also of course therefore at the organismal level. Now, if you want to study physiology, you can't select a particular favorite pathway or mechanism. So what we have done in the lab is actually look at multiple pathways at a systems level, all the way from mitochondria to the nucleus to intercellular communications to extracellular communications. And like Sunil mentioned, we have not left many mechanisms inside the cells. But the advantage that we have in our lab is also to use Drosophila, mouse and humans as model systems, so that we can also address evolutionarily conserved mechanisms that can actually be relevant in the context of diseases. I'll skip this. We study multiple tissues and organ systems, not important. One of the things that we have recently favored as a hypothesis is what really causes diseases. Of course, you do cause disease because of some genetic variations, but often your lifestyle becomes an important determinant for disease onset and progression. So our work in the recent past has shown that oscillations in biological mechanisms, for example, in the context of circadian cycles or feed-fast cycles, if you have aberrant feed-fast cycles or aberrant circadian cycles, they are sufficient to drive diseases. And each biological mechanism has what is called an emergent property. That means if you go from state A to state B, if you accumulate errors in each of these states, they amplify and drive you to a disease state. So that's something that we have established. And these emergent properties actually scale across mechanisms. And we can study at the metabolism level, at a mitochondrial level, at the epigenomics level, at the endocrine signaling level. No matter which pathway you look at it, there is an emergent property. That means something that happens very early in life can set off a series of mechanisms that give you a deterministic output in terms of what diseases you can have later in life. No, but let's say if you are a God, given a truth that molecular biology is all important in terms of understanding diseases, and you vouch by understanding genes to be sufficient to give you diseases. Let's think about how much we understand here. Now, if you want to regulate gene expression, you can regulate gene expression at multiple levels: at the chromatin architectural level, at the transcription level, at the transcript level, at the post-transcriptional level, at various mechanisms. Now, do we know how do the cells choose which of these mechanisms become relevant? When do they choose to regulate transcription at the genome level? When do they choose to regulate transcription at the transcript level? Because each of these have an energetic cost, and the cell has to intelligently choose which of these mechanisms become relevant to regulate gene expression. Now, in this context, I at the last minute changed my slides because I thought I will pick up from where Das had left out. Now, everything that happens in a cell has an energetic cost. I mean, we've completely ignored this. So, therefore, a cell that chooses to regulate something has to balance all the time in terms of whether this is the most efficient way of regulating, controlling cellular physiology. Now, given this, it's important to think about splicing, because splicing is also a very energetically demanding process. Transcription is energetically demanding, translation is energetically demanding, and so is splicing, because every splicing requires ATP to be expended. So we asked a simple question: can we think about how splicing and metabolism are connected with each other, which has been largely understudied or ignored in the field. Now, given this, we went back and asked what is known in the literature about intron retention. And intron retention is an interesting field because all of us know that the genes have introns, and in the evolution, turns out for a very paradoxical reason, higher eukaryotes have many more introns than the lower eukaryotes. And even more weird is the fact that there are more weak introns in humans than in mice, of course much less in lower mammals. What do you mean by weak introns? These are introns which are inefficiently spliced after mRNAs are produced. Now, why would you want to create an inefficient mechanism in the most highly evolved organism whereas you have created a very highly efficient splicing mechanism in a lower organism? It sounds like a bit contrived to me in terms of evolution. But turns out this is very important because you can regulate gene expression at will, and by retaining introns you can do many things, as I'll show in the next slide. Now, intron retention has been shown to be very important for regulating what is called nonsense mediated decay. Now, we know that there could be many stop mutations or nonsense mutations that can happen in the genome. How do you make sure that mRNAs that have stop codons are recognized by the cells? This is a mechanism that keeps, make sure that all these mechanisms are tuned all the time. But it also turns out to be true that when you retain introns, you create new domains in the proteins, and these new domains have new functions. So that means you're creating more diversity in terms of the proteome, which can give you a lot of functional output. Now what we are looking at is retained introns can also act as gating transcripts expression. That means if I want a burst expression of a gene, I can produce transcripts but not splice the introns, so that means they are not translated. When I want the protein to be expressed, I suddenly splice all of them, so there is a burst expression of genes like what happens in a feed and fast cycles, because you want rapid expression of proteins at will and you can't waste time by transcribing them one after the other. You want to make sure there's always a ready-made pool of transcripts that can be immediately taken into action. Now, we simply asked whether can we understand whether there is a crosstalk between energetics and splicing, and we know that a lot of splicing deficiencies are associated with everything from neurodevelopmental diseases to metabolic diseases, diabetes, obesity, and others, because it's been very well correlatively demonstrated. So we asked, can we go back and ask, can we investigate how much of this is true at molecular levels and cellular levels and organism levels? Let's not worry about molecular factors because these are just simply players. If somebody was interested more, we can talk about how this is brought about. So we said, can we simply look at this, what happens during a feed and fast cycles. This is a convenient physiological process because one, you're looking at anabolic and catabolic shifts, which are very important, whether it is for redox balance or for energetic balance. It's also a convenient molecular mechanism because you have to constantly switch the expression of genes all the time. And third, you have huge energetic costs that are associated with each of these processes. Perfect. So we looked at it, and one of the candidates that we specifically chose, although we have looked at whole genome level intron detention, is this particular protein called polymerase gamma. I think many of you would be familiar with Pol gamma. Pol gamma is the mitochondrial DNA polymerase, and in fact several Pol gamma mutations are known to be associated with metabolic diseases and neurodevelopmental diseases. And therefore, Pol gamma expression and levels are extremely relevant for cellular energetics, doesn't matter which process and you think about. So we asked, how does intron detention change as a function of feed and fast cycles? As anticipated or hypothesized, we found huge regulation of intron detention between fed and fasted states, whether we looked at the mouse liver or in the primary hepatocytes or in any other cell type that we have scored for. I'm just giving you snippets of data here. So we found that between ad libitum fed mice or 24-hour fed mice, or if you do mimics of cells using glucagon or fasting conditions, you saw intron detention to change. And particularly for Pol gamma is what we found. So what do you expect? When introns are retained, because your transcripts are not translated, your protein level should be low. Whereas when you introns are spliced out, your translation should increase and intron detention to decrease. So there should be an anti-correlation between retained introns and protein expression. That's exactly what we found for Pol gamma, for example, where we see that whenever you have intron detention high, Pol gamma protein levels are low. When your introns are spliced out, your Pol gamma expression becomes higher. So we found this very well, and we also showed that this is true even if you take what is called a circadian perturbed mouse, where we do what is called a shift worker. We do what is called reverse phase fading. We feed the mouse at the wrong time of the day, and again you can see that there is clearly an antiphasic expression or retention of intron in these livers. Now we have done exhaustive work to identify the causal mechanisms and what could be driving this, and we firmly now have some clues in terms of that all of these regulation is happening also at the level of mitochondria, that mitochondrial energetics is the one that dictates intron retention at a global level. I'm going to skip a lot of these, and in fact we have done a lot of these work to show both causal mechanisms and consequential mechanisms, and it does have an impact on mitochondrial DNA because Pol gamma is directly important or relevant for replicating the mitochondrial genome. So I'll skip this, and I said, okay, now we said this is true. We looked at only one Pol gamma, or maybe we looked at a bunch of genes which are our favorite genes. We looked at AKT, we looked at many other things. But is it true at a global level? Because that's the most important thing. If this mechanism has to have some universal importance, we said, is intron detention regulated at a global level? So we went back and downloaded all the transcriptome data that is available in the literature from mouse and many other organisms, and we said, can intron detention be regulated between fed and fast cycles? And we did find that between feeding and fasting states, your intron detention changes nicely correlating with our hypothesis that is the energetic state that potentially has a global impact on intron retention. If you come up with a mathematical score about when does intron retention change, in fact, if you look at the circadian rhythm, you nicely see an oscillatory pattern for intron retention in the liver, and we found this to be also true to some extent in the intestine, and muscle interestingly shows a similar pattern for exercise-dependent regulation. So all that I'm trying to say is that when you want introns to be retained or splicing to be controlled, you have to think about energetics, because we have thought about energetic cost for every other cellular process. Very little has been shown for intron splicing, and it has important consequences for metabolic tuning. So now, are there some genomic features that actually kind of provides you this bias for introns to be retained? We went and looked at what kind of introns might be having these differential splicing potentials, and clearly something that others have shown, but we have done more work now. Clearly these introns are smaller in size, they have higher GC content, and you can almost now predict at a genome level what kind of introns in what transcripts you can have more retention as a function of metabolism. And if you now look at the entire Go ontology pathways, you can clearly see what kind of genes may show differential regulation. So all that I'm trying to say is that metabolism acts at multiple levels, and these have important physiological consequences. Now, this is relevant because even if you want to think about how transcripts are regulated in a post-transcriptional dependent manner using translational efficiencies. That's what I've shown you earlier. Long ago, about 6 years ago, we also identified what are called hepatic fed microRNAs, which oscillate in the liver between fed and fast cycles. This is the risk-associated microRNA levels between fed, fasted, and refed states. Now, we found that if you perturb these fed microRNAs only in the liver through adenoviral systems, that means you can do tailvein injections of these microRNAs into the liver, either express the microRNAs or scavenge the microRNAs. You can perturb whole body energetics. This is a respiratory exchange ratio for mouse, where only the liver hepatic microRNAs were perturbed, but the whole body energetics was completely changed. Which means that you can perturb these low oscillation molecules to give you a whole organismal phenotype. I'm going to skip a lot of these, and we showed that how this is also circadian dependent, and there is an anticipation for these microRNAs. That means these microRNAs are made and kept in a precursor or a pre-processed state in the starvation state. So that means when you want them to be mobilized, there is processing that happens so that in the fed state, these microRNAs can act on starvation transcripts to regulate fed responses. So I'm going to skip, and in fact we've done a lot of experiments, including by reverse phase feeding the mice, by again making sure that through respiratory exchange ratios, looking at the expression of both mature and precursor microRNAs, and we show that how starvation transcription factors regulate the expression of these precursor microRNAs, but insulin signaling is required for processing these microRNAs. So that means you have both fed and fasted signal cues that lead to this oscillatory microRNA expression. And this is relevant because microRNA dependent therapy is coming up in a major way for many liver diseases, but also metabolic diseases. So the reason I'm saying this is because potentially it's not just cell and gene therapy in the classical way that we think about. Many of these pathways and targets could be used in the clinics to address things. Okay, now my job here is also take the whale level detail, but also at the micro level detail, and see if can we combine the two together. And this is where we've come up with this new center at TIFA called ARUMDA, which is Advanced Research Unit on Metabolism, Development and Aging, which is a very long name, but it kind of encompasses everything that we do in biology. Doesn't matter whether you're somebody who's studying glutamylation of proteins or tRNAs or incretins. All of us can map ourselves into this. Why do we need this? Because there is no dedicated center to study these things in India. And this is not to show, like Nihal said, that I was also young one day. This is just to acknowledge my other co-conspirator called Mahendra, who is an equal lead. Now, the importance here is that ARUMDA is missioned to do something very different. We want to not only do excellent science, but we claim to also empower researchers, technologists, and policymakers. And I'll tell you why this empowering thing comes about. But for all the students, particularly sitting here, when you have a disease that you look at it at in the clinics or in the mouse, if you want to look at a phenotype, you don't know exactly what all sequence of events led to this particular disease. There could be a combination of events that could have led, and in the human context, none of these are happening in isolation from what is the socioeconomic context. So we have to consider both in looking at what causes these disease. And this disease could have multiple origins, or there could be modifiers across lifespan, like what Nihal said. You could have undernutrition during neonatal or maternal undernutrition. Depending on what happens in your adult stage, you can change the outcome in terms of your metabolic fitness. So therefore, we need to constantly think about when disease might have started and how do you track these things. So therefore, you need high clinical definition, you have good epidemiological data, but you also have to combine deep dive biology to address some of these things. Ultimately, my favorite hypothesis is, why does this happen? It boils down to everything that we've discussed in the afternoon session. Ultimately, it boils down to resource allocation, because all the nutrition and the diet you take has ultimately two fates. Either you build body mass, which means either whether it is beta cell mass or muscle mass or adiposity mass, or all of this is used in terms of calories that you need to be burned. How does the body know, how does the organism know when to use how much of calories for building the body mass and how much of calories to be used for burning to give you energy? That is an outstanding question, at least from my perspective in biology, and linking that across physiological state becomes extremely relevant. Now, therefore, what we do in Arumda is use preclinical models, that is mouse models, which are hopefully based on what we think is closely matching what happens in human context, but also we do a lot of human cohorts, and we do what is called forward and reverse translation to tackle this using a large network of people. In fact, this is beyond TIFR, beyond one particular organization, and right now I'm not going to talk about this. This also is something that is very politically important. I will skip all of this. And basically we work with about 40 organizations across the country, and this is relevant because the median age of Indian population is about 20 to 25 years of age now. By 2050, which is the Amrit Kal Bharat that the government wants to really highlight, most of us will be at the age group of 50 years, which is where your normal onset of all these age-related diseases happen. But if you are malnourished, the propensity of the populations to become metabolically challenged will be even further heightened by 2050. So we need to work now. And most of our studies have happened using one or two populations. We need to capture genetic and dietary diversities and also geographical diversities that is present in India. So I'll skip a lot of these, and I'll say why this is, why we call ourselves as empowering. We have established a national analytical facility for nutrition and metabolism research. Right now we are doing a lot of human cohort studies, but soon we are going to be opening this for service. That means anybody and everybody, doesn't matter what type of biology, whether you're a clinician or a basic biologist, you will be able to access the facility to do a lot of work on metabolomics. We are also building human metabolic chambers in CMC Vellore, and hopefully we're going to be inaugurating that soon. And again, thanks to Nihal, Felix and the entire team, we have been able to set up the first human metabolic chambers in the country here. And this is also going to be revolutionizing the way we study these things. We do a lot of cross-sectional longitudinal studies. I'll skip this. We are now spread out across the country in about 8 to 10 sites for multiple cohorts, and again something that I can discuss later if you're interested. With that, I'll quickly go to the acknowledgement sides. None of this would have been possible without a good funding support. Of course, it was a big battle to get this funding, but we've got the funding from various organizations, all the way from DA and TIFR, which was the first to support, and of course several granting agencies, and also thanks to Agilent, because they've also supported by providing free machines for us to set up facilities here. And I would like to thank my lab because my lab pretty much functions without me, and it's I just keep going around from one place to the other, and they do all the work, and thank you very much.
Take one or two questions.
This is with the splicing part. So when you say there is an intron retention, have you looked at the kind of RNA binding proteins or splicing factors that are changing, which is actually leading to this intron retention? Have you looked at that aspect of it?
So there is this what is called polypyrimidine tract which is present in the introns. Now if you have the polypyrimidine tract high, you have strong spliceosome assembly. If you don't have this, the spliceosome assembly is insufficient. We need what are called accessory factors like sun. I didn't describe that. We have identified these factors which have to assemble the spliceosome. Interestingly, the assembly of this spliceosome machinery itself is metabolically regulated, because we found that acetylation of this particular protein, which is dependent on acetyl-CoA levels, you can dictate whether you have better or less efficient assembly of spliceosomes on the weak introns.
So that's my second question. Do you know the signaling cascade that is regulating?
Not yet. We believe acetylation, which is acetyl-CoA dependent and therefore metabolically determined, is one regulation, but there could be several other factors. AMPK could be phosphorylating, TOR could be phosphorylating, AKT could be phosphorylating. I can't rule out any other mechanism. It's definitely possible.
Thank you.
Good question. Yes.
It's a bit of a naive question. So when you reprogram cells to an embryonic state, the mitochondrial state changes. The mitochondria becomes, I think, a bit more immature. So what happens to the intron retention when you reprogram to an embryonic state? I know it's a bit of an off question.
Yeah, so I'm a big mitochondrial fan, but I don't believe there's something called an immature mitochondria, because this is something that people make a big claim based on what happens in a petri dish. In fact, we have understood very little about mitochondrial heterogeneity. Even Sunil, our lab, and several others are looking at mitochondrial heterogeneity. And each of these tissue types and the stem cells behave very non-overlapping manner. So, and becomes even more complex if you think about malnutrition, because everything that we have understood is we have provided all the nutrition to the cells, and your oxygen levels and your mechano sensing, everything is completely artificial. So we need to go back, and mitochondria will be important for your reprogramming, 100% sure.
Okay, one last
I don't know
question.
if I've answered the question. Yeah, so during development, intron retention is the primary thing. In fact, for your axonal migration and crossing the midline, in fact, there is something called robo-slit mechanism that acts. The robo-slit, happens because of intron retention, because the axonal guidance mechanisms go there, and if you don't stop the signal, the axons can turn back and come back to the midline, right? So your intron retention is absolutely essential to make sure that the axons don't continue to respond to the ligand. So they have to cross the midline. So there is intron retention important. During development, intron retention is absolutely essential for gating transition from one cell type to the other cell type.
Good stuff, Ullas. Quick question. So in terms of most of the splicing happens co-transcriptionally, and there's a lot of feedback between transcription and splicing. Do you think, I think most of your talk kind of alluded into these things happen post-transcriptional. Do you think also it's possible there's a feedback and also the chromatin, local chromatin changes or transcription elongation changes with this?
Yeah, so that's a very important question, Pradeep. So basically, most of that has been for strongly spliced out introns. That means if you want to have alternative transcripts, everything from chromatin modifications and all the way to Pol2 CTD that brings about this control. We are talking about weak introns. Nothing is known about how weak introns are related to transcription. We believe burstive expression and weak intron retentions are somehow correlated. We don't know exactly how this is happening. The only possible link is that that protein I showed you, is not only mRNA binding protein, it's also chromatin binding protein. So we believe it is acting as a tether to probably link the two. I don't know. Yes.